Hello and welcome to The Explainer. So today, we're diving into one of the most absolutely incredible construction projects in all of biology, the formation of the human eye. It's this amazing journey of microscopic precision that turns a tiny piece of the developing brain into our complex window on the world.
So this is the real mystery, right? How in the world does a simple little fold in the brain become this incredibly intricate, light-sensing organ that we depend on every single second of the day? Well, the answer is a fascinating story of signaling, folding, and some truly remarkable teamwork between cells.
Okay, let's get right into it. Now, it might surprise you to hear that our eyes don't actually start out on our faces at all. Nope, their journey begins deep inside as a direct extension of the developing brain. You heard that right. The blueprint for the eye is literally brain tissue. And if we want to get really specific, it all starts in a part of the forebrain called the diencephalon. Right around day 22 of gestation, this area develops two little groups, one on each side. These tiny outpouchings, well, they're the very first signal that an eye is officially under construction. Right, so from those simple little grooves, things start to get complicated and fast. The next major phase is all about building the fundamental structure, the chassis, if you will, that's going to house all the eye's critical parts. This is where the optic cup takes shape. The speed of development here is just mind-blowing. What starts as a simple groove on day 22 quickly balloons out over the next week into a hollow little bubble called the optic vesicle. And then, by the end of week 4, something incredible happens. This bubble folds in on itself, a process called invagination, and boom, you've got the optic cup. And this folding action, it's the key architectural move. It's kind of like pushing your finger into a soft, partially inflated balloon. You don't just make a dent, you create this two-layered cup. And in the eye, these two layers, which started as the exact same tissue, are now destined for wildly different but equally crucial jobs. So what's so cool about this single fold is how it creates this perfect division of labor. The inner layer of that cup, that's going to become the neural retina, the tissue with all the photoreceptors that will actually see the light. Meanwhile, that outer layer becomes the retinal pigment epithelium, or RPE, which is basically the essential life support system for the retina.
But get this. This whole thing isn't happening in a vacuum. While the optic cup is busy forming, it's also kicking off another critical project right next door. Because, an eye is pretty useless without a lens to focus the light. And making one requires this perfectly timed microscopic partnership. It's a classic case of what biologists call induction, where one tissue tells another one what to do. So as the optic vesicle grows out, it actually touches the surface tissue, the ectoderm. That contact is the signal. It tells the ectoderm to thicken up, creating something called the lens placode. Then, just like the optic cup, the placode folds inward, pinches off, and forms a perfect little hollow ball, the lens vesicle, that sits right inside the optic cup. It's beautiful.
Now, none of this is random. This whole conversation between the tissues is run by specific molecular messengers, chemicals like FGF and BMPs. You can think of them as tiny little project managers shouting instructions. They're the ones that tell the cells at the back of that new lens vesicle to start stretching out, to elongate and fill the hollow sphere. By week 5, these become the primary lens fibers, forming the very core of the new lens. Okay, so here's the thing. This whole process, this beautifully orchestrated dance, it's also incredibly fragile. The smallest error in that blueprint or any disruption along the way can lead to some really significant and permanent issues. And understanding the normal process is the only way we can understand what happens when it goes wrong. For instance, remember that groove that lets blood vessels into the optic cup? It's called the choroidal fissure. If it doesn't seal up completely, you get a gap called a coloboma. If those same blood vessels don't disappear later on like they're supposed to, that causes a condition known as PHPV. And in very rare, tragic cases, if the whole process just stops very early on, you can have a complete absence of the eye, a condition called anophthalmia. And you can actually see the result of one of these blueprint errors. When a coloboma happens in the iris, it creates this distinctive shape that looks like an old-fashioned keyhole instead of a round pupil. It's a direct, visible reminder of that one specific step, the closing of that fissure that didn't quite finish the job weeks and weeks earlier. And, it's not always about the large-scale structure. That delicate process of building the lens can be disrupted too. If those lens fibers don't end up being perfectly clear, the result is a congenital cataract. And what can cause that? Well, a whole bunch of things. It could be inherited genes, certain metabolic disorders, or even outside factors during pregnancy, like a viral infection. So, when you pull back and you look at this entire complex, fragile, and just brilliant process, you can really step back and appreciate the final result. The journey from a simple fold in the brain to a working eye is, without a doubt, one of biology's most amazing feats. I mean, just think about it for a second. What began as a simple groove in the neural tube becomes, through this perfect sequence of folding, signaling, and cellular teamwork, a complex, multi-layered, light-sensing organ that can capture all the detail and beauty of the world around us. It's just staggering. The eye's creation is really a 40-week journey of absolute microscopic precision. And it just makes you wonder, what other incredible, complex miracles of biology are hiding out there right in plain sight, just waiting for us to take a closer look? Thanks for joining us on The Explainer.